With every trial, every test, we move closer to the rest. A world where pain no longer stays illuminated by brighter days. Waits for a cure. Rise and shine, rise and shine.
Okay. So we just learned that we can have discussions online and offline, so that's a good thing. And you know, yeah, I think Rachel gave an amazing presentation, and that work thing is really pinpointing towards the way we all want to go. Now, as questions raised about efforts, joint efforts, etc., what we also learn is that we need to work much closer together, and both between the continents and within the continents. And I was very happy to see also that that South America has made a unified effort, and in the US we have the MDCs, the Martin Delaney Consortia, which work also often with European sites. We also work with some of these MDCs, but I think the bigger picture would be if we basically can unify efforts done in Canada, the US, South America, and Europe, and maybe also and Thailand we have also. So we have, but that's I think one of the challenges, and one of the things that were there much more during COVID times than what we see today. And I think we really need to do that. But I will talk about the EU2Cure Consortium, so the consortium in Europe. So that consortium, basically was initiated by Charles Boucher, which is a virologist that has been doing lots of work on HIV resistance, was well integrated in both European and US networks. But he unfortunately passed away during COVID times, and he's not longer with us. So he started that initiative with a young doctor, Casper Rox, and the moment that Charles became really sick, he called me, and he actually asked me to mentor Casper, because Casper was a very young doctor, and so from there on we have taken that initiative. Casper has been doing great work, but I'm let's say a bit on the side there, and I try to guide where needed, but the things are definitely moving forward, and he's definitely the engine behind it, and does most of the work. So it's basically the goals are to bring together expertise, to synergize, drive innovation, accelerate development of clinical feasibility, and really to be more in the picture with Europe on HIV cure research. And here you see, and this is not something that happens over one night. You always need to brain massage some people to become part of a consortium. But at the end, we are within 11 European countries. We cover already a huge part. We need to cover even still better some Eastern Europe parts, but we cover both the North, South, Central Europe quite extensively. And for me, most important is that we have the big academic centers and people that think about cure in Europe. They think about the Argus people that have been doing some antibody studies. We in Ghent have mainly doing treatment interruption studies. The people from London and Imperial have been doing antibody studies, and Beatrice Mote in Barcelona has been doing vaccine studies. So, and then you have the Visconti people in France, and I don't want to exclude or say someone is more important, but you want to have at least centers that taking leading positions, and they are all there. So, and basically what you see the centers here and depicted, but what we want to focus on translational research. That means we want to work together, people living with HIV, not only, let's say, complex mice models, but starting from the people and translate that to the bench, and from the bench back to the people. So that means we need cure cohorts or people that start to control We need to further analyze what Rachel said, you know, these CD8 biomarkers, assay development, and do interventional trials. And a key point is obviously is the community, and that I think has been recognized very well, but still need to improve, and especially on the fact that I feel that what we see in the study in Thailand, what we see also in the study in San Francisco, you know, what we saw in our treatment interruption study long time ago, where we want to look at the rebound, where is rebound is coming from. We do our very, 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 very best to have one woman included, and basically that has to change. And I think it's been very good discussions on that. I'm very grateful that we have that discussions also. So here you see all the people within the network, and I think that's really a very nice, you know, picture showing that people really want to collaborate. And if I want to mention two people that are driving this, sorry.
You can point it. You just drag it over. Let's see.
Okay, yeah. So we have here Sarah Valk, which is a project manager, and on completely the other side. So we have a project manager in Ghent and in Rotterdam that help driving it, and Casper Rox, which is, you will see, if you see him once, you will never recognize, he has this big curly hair. And he's the young physician. Really, I must say, you know, he's a really an inspirational guy, because he's bringing everybody together, and that there's been efforts for many years by many people. But I think, you know, also by the fact that he was not that established researcher, but someone young that might have facilitated. But at the end, I think we are very grateful that he's there. And we have the people that had, you know, in Charité, Christian, who had that second Berlin person that was cured from HIV. We have Javi Martinez Picado that has from the group from he's here that has led the iSystem cohort that looked at all the people that have been cured. So we're really grateful that we have lots of good scientists there. And we also have EATG there, so the European AIDS treatment group. I think they should change their name to HIV treatment group, but that's my personal opinion. But we have George Barbakeshi there, he's really the person that is helping driving EATG work. Okay, so what do? We have obviously basic science that we want to do together. So create a collaborative research pipeline that informs clinical trials in real time. So much more than what we do now, it's where we really have a lot of post hoc analysis on small numbers, and that's always like bringing it more, making it more difficult. We also want to streamline much better the trials and do that under a common operational framework, so that samples are taken from the same, let's say, with the same operational procedures, the same way, the same number of biopsies that you use for this and this, so that we can start comparing outcomes of studies, because that's one of the big things that we don't have today. And we also working on a Marie Curie doctoral network. So that is a network where we train new people, young people, to become the researchers that search for a cure within 5 years or within 10 years. If we don't have a cure yet, that we have very well trained researchers that can help us doing that. So basically what Céline Gubster is leading is an effort to do this basic, to establish really basic research pipeline. And that's really looking at the clinical trials and their design, but also on mapping all the assays and what is the expertise of the people, and what are the materials that are available. So we have, for example, in some clinical trials, we can have NHP models, but let's say from clinical trials in people living with HIV, we have PBMC, so blood cells, but we also have from some people gut, lymph node. So we are currently mapping the assays, so the technology, and so to go to the next step where we do this more integrated analysis. So just another word on this Marie Curie network. So the aim of these networks, so that is our networks existing in Europe, which have really the goal to train people. So it's basically you want to, the aim of our network is really to have a team that works and is trained to work on HIV cure, and do that in both an academic and non-academic sector. So you train people that work both in companies and in the academic sector to have that broad view how to do research. And so we are right. So that program is coordinated also by Beatrice Mote, who you see here on the right, which is the person that has lots of experience also with treatment interruption studies and vaccine development. So that network we really working hard on. And in general, if you look at EU2Cure, let's say, what are the short and mid and long-term objectives? Basically, what we want to do, and I think sometimes we say it's very let's say autocratic or so, but we have very tight GDPR rules in Europe, also biobanking rules. So we need to have, if we want to work together and we want to do that and exchange sample, we need to have to consolidate across European legal data, clinical, and also infrastructure network. So that's what we're doing now. We're really working on this to have all these institutions agreeing on, how can samples be safely exchanged, etc. We have this Marie Curie network, and we boost EU2Cure visibility, and we also interact with the European Commission. So that's for the first. That's what we are doing right now, and then in the next 3 years, we want to embed HIV cure research more into the scientific agenda of Europe, because Europe has been, over the last 10 years, if you look to focus points of research, it's much more on elderly diseases, dementia, Alzheimer, Parkinson, cardiovascular disease, cancer. So they had a very strong focus on that and less on infectious disease. COVID has changed that perception, and that momentum we want to use to really put HIV back and more pronounced on the agenda. So we want to secure sustainable funding and allow the consortium to grow. We want to prepare for a landmark trial, so multi-country trials. That's what we are doing now, and we do that together with Horizon Europe opportunities, but also a bit independent of that, because we don't want to depend completely on European funding alone, or funding from the Commission, let's say. And then on the long term, we really want to proactively change the way we're looking at cure strategies and train future leaders in cure research. So what do we do today? So we have this Rio trial, which is a study very similar as what Rachel also said in this study in South Africa. So also in Europe, in London, there's a study where people look at the effect of broadly neutralizing antibodies in people treated during acute infection. And that study initially was starting off only in the UK, and then Denmark was added. And now the Netherlands, Spain, Belgium have all been added, and Germany as participating sites. So we closed the inclusion because everyone could have some people that participate. So that's one thing we do together. Then in Ghent, and Rachel has been there, Steve has been there several times, we have an EU2Cure symposium. So I started that 10 years ago, and now we officially made that the EU2Cure symposium. So that's a symposium of the network. And we have also a workshop in June often in Aarhus, where young researchers present their work on a very interactive way. So that's to disseminate research and do some networking. So we have a study that started last year where we built that legal framework. It's called EU2 Control. I will say something about that. But basically, the pillars are that we want to build a PTC biobank, post-treatment control, involve the community from early on, also to have some questionnaires with a virtual platform, and I come to that also, do some bioinformatic modeling on the interventions that has been done. So the EU2 control study is the first, let's say, broad study that runs over all the trial, over all the centers, but what it's basically doing is in the past, several centers have done treatment interruption studies. We just had this paper that also was referred to, where we look when do people in general rebound, and now we look at safety in that group, but also we bring together the biobank samples under one umbrella to make sure to allow to do more analysis on the blood samples that are available. So it's really to check what are the drivers and how can we identify biomarkers that predict rebound and also that predict control. These are very important questions in the field. So what we, instead of working in individual sites, we want to improve by having one clinical cohort where we look in that consortium to post-treatment and post-intervention controls with cutting-edge technologies and with communities, people from the community involved, because people from the community also sometimes attract other sites or can help us identifying post-treatment controllers, etc. So they are very, very closely involved. So basically, we can say that, viral reservoir, there has been people that believe in it, people that don't believe in it, but also in the study that was just presented, the viral reservoir did not really distinguish and some comparative protective factors from before can also not really always distinguish between controllers and non-controllers, but it's more about, the number of intacts, if they are very low, that can be something, and also a strong anti-HIV immune response that what is now called these naive T cells with the stemness, for example, that is something very important, and that can help us. Also, how long has someone been on ART, and also the period between the diagnosis and the start of ART. We did not discuss that, I think, in any session today, but the virus, what we discussed was the virus is always changing, right? It's can make many mutations. So people in which the timing has been longer between the diagnosis and the start ART have a much more diverse reservoir. And that's more difficult to target than people which have been diagnosed and start ART within 1 year, for example. It's not only the reservoir that is smaller, it's also the diversity that is way smaller. Okay. Oh. Did I do something wrong? They all go?
You slid this way. You don't slip on the side.
Yeah.
So all you need to do is hit this.
Yeah. It's okay. It's okay. Sorry for that. Everyone is awake again, right? So yeah, I always do that in the middle of my presentation. Now, so basically, I think that we have discussed. So that this is all about bringing the samples together in a virtual network study and look at the post-treatment control. Now, you can say, okay, but that's not so difficult. You just bring it together, but it's really, establishing this biobank, being able to exchange all the samples. I mean that, and between more than, if it's only between two centers, you need what we call a material transfer agreement between the two centers. But if you want to start, you do the plasma and do this analysis, and you do on these cells this, and you do on, so looking really at key expertises of centers, then your material transfer agreement has to kind of being an overall umbrella-like agreement, and everyone has to agree, and that takes a bit more time. But okay, we have that, and we then in a second phase we expand or post-treatment control biobank to and every site is then functioning as a kind of a mother site with sites around him which can collect post-treatment controllers. And then we go to 42 clinical sites in Europe. And then we have to do all the analysis still in the lab, of course, but we are moving now, we're nearly finishing phase 1. Okay. That I think I discussed. So another thing is, how to involve more the community. Though we have been working on a questionnaire also together with members of the community that will be online available on how they perceive cure, what is our the biggest needs, what should we change in the way we design our studies, what should we change in terms of inclusion or exclusion criteria, how can we adapt or informed consent, etc. So can we improve on the dissemination? What is the role of EATG in general? Can they be more supportive? Because EATG, they have been mainly active in Europe, so the European AIDS Treatment Group, in making sure that everyone got access to drugs. But they have not been as active, I think, as, for example, groups in the US on the cure agenda. So that's something we really need to take up. So that these are all things that are out now, and this www.EU2Control.com, that virtual platform where we will list all these things. So and here you see, we definitely want to improve on community voices, and not only in trial design, but also in inclusion and in communication, pre and post, etc. So and I think what I already said, if we look at the inclusions, one of the things is that some sites really are mainly only having gay men. So that's one thing. So if you look at the Aarhus sites, in Aarhus in Denmark, the number of people in their sites is like 85% are men. We are in a different situation. So Belgium, Ghent, we have about 30% women and 70% men. If you look at this 30%, you basically should look at it differently. We have 50% white gay men, and we have 50% that are people that come from other countries. And this the last 50% is divided in twice, equal men and male and female. So basically, Belgium has some, also not to be proud of, but some history with Congo, Zaire, and with Rwanda, etc. And therefore, I have lots of people, French-speaking, let's say, people from Congo, Zaire, and from Rwanda that I take care of in my daily practice. So we need to definitely to improve on that, and because every year I have an information evening with the African community. So I have a good connection with them. But, you talk about this is information about HIV. And I do know that it's important for them. And when I go there, I see some of my patients are there, but we never talk about, they not openly can say that they are HIV positive. So that's still also for the African community. And something that we also have to think about how we can tackle this. Okay, so the biobank, I already explained about that. So basically, we want to start with having about 100 people that are post-treatment controllers, and then increase that number. What do we do? Well, there's nothing, let's say, very special, but you look at the reservoir, all these immunology parameters, cell function. We already hear that NK activity, the T cells, the B cells, which proteins are involved. So but the technology has evolved massively. You know, the way we do now, the analysis of samples compared to 10 years ago, when I kind of also started the lab, it's mind-blowing changed. So it's impossible to compare that. It's also much more expensive. And that's one of the things we face. You know, we have these samples, several time points. You know, we need to make a puzzle that is complex, but okay. So that I think I can skip that. So what is on the horizon? So we have, so as I already said, we did that study where we looked at viral rebound, and now in the same group we want to look at the long-term follow-up of people that underwent an analytical treatment interruption. Why do we do that? Because, you know, we always say short-term treatment interruption seems to be safe, but that is, you know, that wording seems to be safe, right? We cannot say that 100%. And we know that from other studies, we know that high viral loads drive inflammation, drive also to a certain extent cardiovascular disease. So, and the way we look at it and we think that treatment interruption studies can be done safe is because we don't want them to be too long. But you hear, yeah, too long, you can hear what is too long? Is that 8 weeks? You know, and we don't have all the tools at hand to say that there is for 100% sure that we're 100% sure that there is no harm. We have it to a big extent, but we really want to rule out by looking at these 300 people, and we have a matched group on age where we want to look at development of cardiovascular disease. So that's ongoing and led by a team of Sarah Fiedler, which is a close collaborator in London, in Imperial. And then we have follow-up studies. So the Rio is already ongoing, that I already explained. And then we have studies within different sites in Europe. So the IDEN study is a study led also by Ole Sogard with a low dose of an immunomodulator and broadly neutralizing antibodies. And then there are some vaccination and latency reversing agents. So this is all ongoing. But I really think we should do efforts to make sure that the outcomes are measured equally, that we can cross-validate or use the endpoints equally, like in both with the US, Canada, South America, etc. studies. So I'm gonna close here. So you see all the sites. I think as we saw also for South America, I think there has been some wake-up calls and some very motivated people that start bringing people together, and that should amplify the moment that we can move forward really towards well-designed interventional studies with controls, etc., that take us to the next steps. And I'm convinced that if we look to all the data, we live in a completely different era than 10 years ago. And I also always, because people ask me that many times, do you think we made progress, and why do you think that? And then I say, you know, between 2008 and 2018, we only had one thing basically to do, and we tried to activate the virus and activate, it's called shock and kill, and everyone was talking about shock and kill. And at the end, we realized shock and kill did not work. You know, it might be a component still in the future trials, but it's definitely not going to be the key element that we thought it would be. So that we learned, that shock and kill will not work on its own. Another thing is, in the meantime, between 2008 and 2018, the cancer fields jumped with immunotherapies. You know, they made really success that changed the life of people, that changed from going dead to being alive. And then we jumped in with, you know, these immunotherapies like Rachel did. And that is really, for the first time, we see people that start control. Not a lot, but we see if between 10-15%. We will show data at the meeting when we treat people living with HIV with anti-PD-1 and alpha-4 beta-7, where at 6 months, 22% of the people have a viral load still below 1,000 copies. You know, this is learning us that we're making progress. You know, we can change that curve, but we are not there yet, and we have to work on it. But it gives hope to the community, but also to the researchers, because I hear the question about vaccines, and at a certain moment also researchers become desperate, because if you never have a sign after 5 years, you say, okay, maybe we need to do it different and harder and work more. But after 15 years, you know, so you need to have positive signals somewhere, also for the funders. So this is a key element, that we make progress, that we go to these well-designed trials, where I hope EU2Cure can be a partner with the other continents, and that we can do that together. Thank you for your attention.
Our last speaker for the day. So, any final questions that you have?
I'll bear to you, because they're all really
I actually have a community related question to thinking about like a European consortium. So do you have cabs sort of within the individual countries, and how do they coordinate with each other? And I would presume there are differences in priorities in different places, as you already mentioned, like populations are really different and people's lived experiences are really different. And so sort of like, how does the coordination occur, and even a language barrier?
Yeah, so many of us, sorry.
Repeat your question for the
Yeah, the question is, excuse me, the question is, how do the CAPs function in different countries, and how are they connected to each other? Basically, they are not yet connected. And we start to connect them. I really think they are all connected to CARIN-DB without knowing each other, you know? That's the truth. And we have to improve on that. But I think she's really the link for the moment. Yeah.
Dr. Linos, what can you tell me, if anything, about your surveillance data in reference to newly diagnosed individuals within Europe? Because one of the thoughts that just came to my head again in reference to your network, right? You're working with different countries, different institutions.
Countries, yeah.
Countries, right?
Yeah.
So there are different countries. I'm actually thinking, are any of you, as the researchers, looking at the earlier we get individuals who are newly diagnosed, you know, this can be an engaging point for people newly diagnosed, getting the information in reference to the quicker, you know, you work together with us, you know, it may impact you either curing your disease, you know, to give them that hope of you newly got diagnosed, but there are some things that we can do within the research that might just be of help to you. So do you have any?
Well, what we do have is we have a program, Aware HIV, where people like where we try to bring down the let's say the bar for testing to improve diagnosis, but we have not linked them. Maybe we should link them. Yeah, better. Yeah, yeah. No, I think it's a good suggestion to link them to EU2Cure. Yeah.
Quest for a cure, rise and shine.